Hello everyone, myself is Dr. Avinash Ammangi. I am from Gurudev Academy, Dharwad. So today we are going to discuss with respect to the synopsis of microbes in human welfare chapter, Karnataka Common Entrance Test, right? So with respect to the K-CET only, briefly we'll go ahead with respect to this particularly what chapter, that is microbes in human welfare, right? Now, if you consider overallly what all parts or what all topics we are going to cover up with respect to this particular chapter, that is microbes in first what household products, then microbes in industrial products, then microbes in what sewage treatment, microbes in biogas production, and microbes as a biocontrol agent, and even microbe as what biofertilizer, right? So we'll discuss one by one with respect to this particularly topic. Now, various microbes, right? Various microbes with what different shapes, size, what you are going to find out, which even sometimes we cannot see with respect to our naked eyes. But when they form a colonies, we can see this particularly what the whitish milky type of what colonies. Majorly, if you consider bacteria as of what different types of bacteria we have even studied, right? One is like cocci, bacillus type, like, or even spirillum type, that different types of what bacteria what we are going to observe. So here they have given something called as what bacteria with a what rod shaped, that is what we call it as bacillus. Then spherical shaped bacteria you are going to find out, or spiral shaped bacteria. Spherical shaped means what we call it them as cocci, right? Then spirillum, like what it has a many flagella with respect to that, right? So that what you are going to find out. So different types of bacteria with a different shape and size what we are going to observe. Then again you consider bacteriophages, which is known to be the virus which causes disease to the what, the particularly disease or infection to the what bacteria. Then adenovirus, which causes mainly like what respiratory infections consider. Then rod shaped tobacco mosaic virus, that is what we call it as TMV. These are the what the structure what they have actually shown here, right? Now, as like consider some of the bacteria forms what colony. When you prepare something called as what a media, nutrient media, when you are giving it a artificial medium to grow, then definitely you will find out what whitish type of what colonies like this growing over all over the plate, right? Mainly with respect to the what petri dish what we call it as. Then fungal colony you are going to find out with respect to the what petri dish only, right? There also like what we are going to use different nutrient medium. Majorly like what, just for a knowledge consider, this particularly fungi can be grown over with respect to the what PDA media, which is nothing but known to be the potato dextrose agar, right? But here if you find out, we here with respect to the what bacterial colony, we call it as what Na media, that is nothing but what nutrient medium or nutrient agar medium, that what we are going to find out with respect to the growth of this particularly bacteria as well as what fungi, right? Then you consider like what microbes in household product. So one by one we will discuss with respect to the what household product. Majorly like what lactobacillus bacteria or lactic acid bacteria, what we are going to call it as, majorly known to be commonly we call it as what lab, right? So lab, if you find out like, that is lactobacillus, which is now names is what bacillus means what the shape of the bacteria will be what rod shape, right? Then majorly what it do, it converts the milk into what curd by producing something called as what acid, and that coagulate and partially digest the particularly what the milk protein, right? Now what this is going to do, the lactic acid bacteria is going to partially digest the milk protein and convert that milk into the what curd, which is what coagulative form of what milk, right? Then this is due to the what the production of what the particularly lactic acid by means of what fermentation method only, it converts that that milk into the what curd form, right? Then fresh milk can be what converted to curd by adding some of the what curd containing lab, right? And it also increases something called as what vitamin B12, right? And in stomach, if you find out, the lab helps to what check the pathogens, right? Checking of the pathogens, like what even lab we call it as probiotic bacteria, because it is going to help us to check the pathogens again, and even it will help us something called as what carrying out a different digestion activities, right? So the because of that only, like what in Indian food items we prefer to have this particularly curd, right? After our what lunch or dinner we actually have this particularly curd, right? So which actually increases what vitamin B12. Majorly what is going to happen, conversion of this particularly curd from the milk to curd is by means of what fermentation method. That is what you have to remember. So commonly found out, what you are going to find out. Then consider like what the particularly bacterial fermentation, majorly like what anaerobic type of what respiration, right, in which the duff is what used to make something called as what food such as dosa, idlis, consider, right? So to prepare a duff, like what they do is they have this particularly all different types of what stuffs and add up this particularly what bacteria. Then later period what in the morning you will find out the growing of this particular duff, and the duff that can be used for the preparation of a dosa and idli. Then puffed items, like what mainly appearance of what duff is due to the what the production of what CO2 gas. This is what you should remember. So puffed stuff what you are going to find out. So as it is going to be puffed, so definitely there is a production of what CO2 gas in it, and that is what you can find out with respect to the what fermentation activity only what you are going to get. Then again there is something called as what baker's yeast, that is Saccharomyces cerevisiae, what we are going to call it as, right? It is used to make something called as what bread by fermenting duff, right? So again they will prepare a duff, with respect to the duff only, like what fermentation takes place, and fermentation basis we will find out bread even what spongy. So there they use this particularly yeast, which is nothing but a unicellular type of what fungi. Scientifically it is called as what Saccharomyces cerevisiae, right? Then again majorly like what the different products what we are going to discuss, like toddy you consider, then fermented fish you consider, then fermented bamboo shoot you consider, then fermented soya bean you consider. So there also what they are going to use the different kinds of what microorganisms to carry out what fermentation. So toddy is actually made up of what fermenting the sap from the what palm. So from the palm tree only what they are going to do, they are going to remove the sap and they'll undergo something called as what fermentation activity. Then microbes are what used to ferment the fish, even soya bean and bamboo shoots, even to like and to produce what particularly cheese. So for the production of what cheese also we are going to use something called as what microbes here, right? So which actually carries what fermentation. Then even there is something called as what Swiss cheese and the Roquefort cheese you consider. Majorly like what Swiss cheese has what large holes due to the what production of what CO2. This is what you should remember here. So Swiss cheese has what a large holes due to the what production of CO2 by the majorly bacterium name you should remember, that is Propionibacterium cermeni, right? Propionibacterium cermeni is a bacteria that is nothing but what which will help in carrying out the fermentation of this particularly Swiss cheese, right? Then again Roquefort cheese if you consider, which is nothing but what ripened by growing up what fungus, mainly like what Penicillium roqueforti on them, right? Majorly like consider ripened form of what whatever the products are, mainly like what fruits you consider. On to that we are going to grow up with respect to this particularly Penicillium fungi, and which carries out something called as what fermentation, and with respect to the fermentation only we can even isolate this particularly what cheese. Then microbes in what industrial products, majorly like what this is what the most important aspects even you can consider, because with respect to the development of what biotechnology, and majorly we have already studied with respect to this particular chapter known as what biotechnology principle and process, that majority of the microorganisms can be used into the what even food industries, so which can help in to produce what different kinds of what products, and that products are what useful for the what human beings, right? So you consider how they are going to carry out, they are going to carry out by means of fermenter, that what even we have discussed in biotechnology principles. So based on the fermenter or based on that particularly big vessel only, they'll undergo with respect to the what fermentation procedure. So majorly here if you consider by means of what fermentation, production of what beverages, antibiotics even you can consider on an industrial scale, majorly like what large scale, requires what growing the microbes in a very large vessels, and those vessels we are going to call it as what fermenter. In a large big vessel only they are going to grow up this microorganism which carries out the fermentation activity and will help in production of what different beverages and even antibiotics, right? So here mainly fermented beverages, if you consider like what Saccharomyces cerevisiae, mainly even it can be known to be brewer's yeast, what we are going to call it as, to prepare different beverages. They are going to use this Saccharomyces cerevisiae, right? And is used for the production of what beverages by fermenting the particularly malted cereals even. So cereals we are going to take and we'll go ahead with respect to the malting and carrying out the fermentation, and which will give me something called as what beverage. So that is what wine and beer if you consider, right? Then from the word fruit juice to produce what ethanol. To produce ethanol means what definitely their Saccharomyces cerevisiae is going to use the carbohydrate present inside these particularly materials, and which will convert that into what ethanol. So for the preparation of what wine and beer, we are going to use something called as what this particularly yeast. Yeast as a fungi, mainly we are going to call it as Saccharomyces cerevisiae, or even we call it as brewer's yeast, right? Then wine and beer are produced without what distillation. Actually there will not be any distillation procedure over here. So directly even it can be consumed. But if you consider whiskey, brandy, rum, gin or arrack are the produced by means of what? Distillation of what? Fermented broth. So after the fermentation they are going to collect that particularly fermented broth, and then they will go forward distillation procedure, because the concentration of alcohol or ethanol will be very high with respect to the what whiskey, brand, rum, right? So definitely like what to dilute it. For the dilution you consider majorly like what purification, even what we call it as the distillation procedure is carried out, right, with respect to the what using fermented broth. Then there is a production of what antibiotics even, right? Majorly these antibiotics are what chemical substances produced by some of the microbes and can kill or retard the growth of what even pathogens. So some of the like what we call it as, in Hindi we say one quote that loha lohai koi kaatta hai. So it means same way only like here we are going to use the microbes for the production of what different antibiotics to treat the microbes majorly which are known to be what pathogens. So to kill that particularly pathogens we are going to use this microorganism itself, right? So used to treat mainly like what plague you consider, whooping cough you consider, then diphtheria you consider, leprosy. For that purpose we are going to prepare something called as what antibiotics by using the particularly microorganisms, right? Then again if you consider like what antibiotics, right? So penicillin is the first antibiotic known, right? The first antibiotic was discovered by whom? Alexander Fleming, right? Now what he observed that the particularly staphylococcus is a bacteria could not grow around a mold. Around a mold, which mold? The Penicillium notatum, right? Mold means what? Fungi, right? When fungi, this particularly Penicillium notatum started growing, right, with respect to the what surrounding, there will not be any growth of this particularly staphylococci, right? Then growing in unwashed culture plates, he extracted what? Penicillin from it, right? Then what he have done? He have taken this particularly Penicillium notatum, grown it, and unwashed culture plates he have taken, and from there he extracted something called as what? Penicillin, and that penicillin is known to be the what? Antibiotics, and it is the first antibiotic discovered by whom? The Alexander Fleming, right? So that what you should remember. Then again Ernest Chain and Howard Florey established its full potential as an effective antibiotic. Then later period like what the majorly like what penicillin can be used as what? The antibiotics. So for that purpose this particularly Ernest Chain and Howard Florey played what very important role. Then Fleming, Chain and Fleury were awarded what a Nobel Prize for this in 1947. Right after the discovery of what penicillin extraction they have done, and later period these two of them, Fleury and Chain, established like what full potential of this antibiotic against some of the what pathogens. Then they awarded, these three of them were awarded with the what particularly Nobel Prize in 1945. Right. Then some of the like what chemicals, enzymes and other biotic molecules can be bioactive molecules can be produced from such kind of what microorganisms. So here something called as what Aspergillus niger. Aspergillus niger is what, it's a fungi, right? Now you consider this is a structure of what the Aspergillus niger. Majorly Aspergillus niger is used for the what production of what citric acid. So these are the what component what we have to remember. So citric acid is produced by means of whom? Aspergillus niger, or Aspergillus niger produces what? Citric acid. So that what you should remember. Acetobacter aceti, which is nothing but a bacterium, right, which produces something called as what? Acetic acid. So name itself will tell you that it produces something called as what? Acetic acid. So Acetobacter aceti, that is nothing but it produces what? The majorly acetic acid. So here you can find out what? Acetobacter, right? This is the structure of what particularly Acetobacter. Then Clostridium, you consider Clostridium botulicum, which produces actually what? Butyric acid, right? So butyric, right? So like that what we can remember. This is even known to be what? Bacterium, right? This is also the botulicum is even known to be what? The particularly rod shaped type of what? Bacteria. Then lactobacillus bacteria, which actually produces what? Lactic acid. So that is what the structure of what? Lactic acid, or sorry structure of what? Lactobacillus bacteria you can find out. So which actually produces something called as what here? Lactic acid, right? Now again if you consider like what? The alcohol, for the production of alcohol what they are going to use? Yeast. That is nothing but what we call it as Saccharomyces cerevisiae is used to produce something called as what? Ethanol. Structurally it looks like this, right? But you can get it into the form of what? Powder form. So what you have to do, the dried or immobilized powder, that what you have to add it into the water. When you add into the water, the cells will become what? Active, and then it can be used for the what? Fermentation procedure, right? Then enzymes, you consider some of the enzymes even can be used for the what? Different purposes. And now these enzymes are actually isolated from where? From the microorganisms only. Majorly lipases if you consider, which is used in the what? Detergent for formulation and help to what? Remove the oil stains from the laundries, right? Now what lipases does? It removes the oily stains from the what? Laundry or from our clothes, right? And which can be used with respect to the what? Detergent. While detergent formulation, during that period they add something called as what? Lipases. So these oily stains what you are going to find out onto the clothes, that can be removed by means of what? This particularly lipases, right? Then there is something called as what? Pectinases and proteases to clarify something called as what? Bottled juices, right? So with respect to the bottled juices, to preserve or to clarify it, what is required? Pectinase and protease even is added with respect to that, right? Then again you consider streptokinase, majorly like you consider which actually produced by means of what? Streptococcus bacteria and used as a what? Clot buster. So streptokinases is an enzyme which actually help in what? Like a clot buster you can find out, right? Which actually remove the clots from the what? The blood vessels of the what? Patient who have what? Myocardial infarction, right? So myocardial problem if there is there, then definitely to what? Burst that particularly clot from this particularly a patient, that time they use what? Streptokinases. Now this streptokinases is produced by whom? The streptococcus bacteria, right? So that what you should remember. Then there is like cyclosporin, consider, which is even a drug, right? So cyclosporin what we are going to find out? The capsule form, right, produced by means of whom? Trichoderma polysporum, right. So Trichoderma polysporum, which is a fungi. What it is? It is a fungi which produces what? Cyclosporin, and this cyclosporin used as what? Immunosuppressive agent, right. During what? Organ transplantation, or the patient who undergo organ transplantation, they actually required what? Immunosuppressant, and that immunosuppressant should be taken throughout the life, right. So for that purpose, like mainly Trichoderma polyspermum itself, polyspermum produces what? This cyclosporin, which will act as what? Immunosuppressive agent, right. Then stannine you consider, which is actually produced by means of what? Monascus purpurus, right. Monascus purpurus, which is known to be what? Again, yeast, or the particularly fungus, right, used as what? The blood cholesterol level lowering agent, right. So this can be used as what? Blood cholesterol lowering agent, and it inhibits the particularly what? Enzyme responsible for the synthesis of what? Cholesterol. So stannine, what it does? It actually inhibits the enzyme which is actually responsible for the synthesis of what? Cholesterol. So only we call it as what? Stannine can be used for blood cholesterol lowering agent, right. Now this stannine is actually isolated from where? It is isolated from the Monascus purpurus, right. So from that particularly what? A yeast, or a particularly what? Fungi they produce. So you should remember these names. Stannine you consider, which is actually isolated from Monascus purpurus, right. And this stannine is used for what purpose? Blood cholesterol lowering agent. So these all things what we have to remember with respect to the what? CET concern, right. Then we'll go ahead with respect to the what? Microbes in sewage treatment, right. How these microorganisms are going to help in the treatment of this particularly what? Sewage, right. Sewage means majorly like what? Contaminated water. For that what actually required. Now you consider sewage, mainly what? Municipal wastewater it is known as, contains what? Large amount of what? Organic matter and microbes. It consists of large amount of organic matter, and even lots of what? Microbes can be can grow over with respect to that. Then sewage is treated in sewage treatment plant, right. Majorly we call it as STPs, right, which is nothing but sewage treatment plant, to make it what? Less polluting. So less pollinating. For that purpose we can go ahead with respect to the what? The particularly sewage treatment. So there are two stages. One is known to be the primary treatment, and one more is known to be the what? Secondary type of what? Treatment. Even secondary type of treatment can be known as what? Biological treatment, where again we are going to use what? Microbes. So primarily what they are going to do with respect to this particularly sewage, that what we will understand. So in primary treatment, it is what? The physical removal of what? Particles. So physically they are going to what? Remove the particles. It includes mainly what? Removal of what? Floating debris by what? Sequential filter. Then removal of what? Grit or soil or small pebbles by means of what? Sedimentation. So what they are going to do? They are going to pass the water, this particularly contaminated form of what? Water can be passed from here, and where the larger particles is going to pass on with respect to the what? Sequential filters, right, with respect to the what? Pore size. The large particles are what? Separated from it. Now who is going to get enter? The grid chamber, what we call it as. Majorly grid chamber means it consists of something called as what? Grit means what? Soil and the particularly small pebbles that are actually collected over with respect to the grid chamber. Then it should be passed on with respect to the what? Sedimentation tank, and then the next like what you are going to find out, the carrying out the secondary treatment, right. The majorly like you consider the settled solid forms, like primary sludge what we are going to call it as, and the what? Supernatant forms the particularly what? Primary effluent. Now whatever the water is passing on, we call it as primary effluent, and whatever the settled or solid particles what actually separated from this particularly primary treatment, that what we are going to call it as primary sludge, right. That what you should remember here, right. Then in secondary treatment, what they are going to go ahead, even secondary treatment, what it can be called as what? Biological treatment, right. Now what they will do, whatever the primary effluent was, right? That primary effluent is passed into the what large aeration tank. So with respect to the large aeration tank, that what you can see here, right? With respect to that, they'll pass on with respect to the aeration, the microbes even can be what grown up. So what they will do, constantly they'll agitate this particularly what water. Now after the particularly what agitation, this allows what the vigorous growth of what useful aerobic microbes, right? Majorly what you are going to find out over here, in primary effluent, whatever the primary effluent, that is actually passed into the what aeration tank, which actually aerate this particularly even the effluent. And when they aerate, with respect to that aeration only, you will find out what grow of large amount of growth of what aerobic microbes you are going to find out. And these microbes are going to grow up into the what flukes. Now what these flukes? Flukes are nothing but it's a bacteria associated with respect to whom fungal filament to form a mesh like structure. Now what fluke is? It's a bacteria which is associated with respect to the fungal filament and forms something called as what mesh like structure. That what you are going to find out here, right? Once these microbes consumes the particularly organic matter in the effluent, this reduces something called as what BOD, that is nothing but what biological oxygen demand of the effluent. Now what these microbes are going to do, these microbes are going to utilize the organic component present inside the water and makes the water what purify, and it actually reduces what BOD. BOD is nothing but what known to be the biochemical oxygen demand. Now whatever the chemical, organic chemical was there, that actually used to utilize something called as what dissolved oxygen present inside it. Now what we made it with respect to the flow only, we actually reduced this particularly what BOD, that is biochemical oxygen demand. Then mainly you can consider these are the what flukes what you are going to find out. It is nothing but a mesh like masses of what bacteria and even fungal filament. That what you should remember, that is known to be the primary treatment. Now with respect to the what this biochemical oxygen demand is, right, what it is, it's an amount of what oxygen consumed by bacteria to oxidize all the organic matter in a 1 liter of what water. Majorly what it is, amount of oxygen consumed by bacteria to oxidize all the organic matter in 1 liter of what water. That itself what we call it as BOD. It measures of what organic matter present in water. So actually what it gives me, it gives me something called as what organic matter present inside the particularly what water. The greater the BOD, more is its what politic potential. So how much the like what BOD is, if consider if BOD is more, then mainly like what water is polluted one. That what we can consider here. Then in secondary treatment, like majorly whatever the effluent is, that is what treated with aerated and flux, right? Then the effluent is then passed into the what settling tanks. It is actually passed with respect to the what settling tanks, what you are going to find out, right? Where the bacterial flux are flux what sediments, and this sediment is called as what activated sludge. They'll again go ahead with respect to the what the where the particularly bacterial flux are sediments, and these sediment is known to be what the particularly activated sludge. Again there is utilization of what microbes which actually purifies this particularly water by using the organic and inorganic substances present inside the water. A small part of what activated sludge is pumped back, right, into the particularly aeration tank to serve as what the particularly inoculum. So what they'll do with respect to this particularly activated sludge, they are going to pass on those that particularly small amount of water to the what aeration tank. So we can call it as what something called as what inoculum, right, which majorly inoculum means which majorly consists of this particularly fluke or a bacteria which is actually associated with respect to the fungi, filamentous fungi, right. Then again you can find out here the remaining sludge is pumped into the what large tanks called as what anaerobic sludge digestion, right. Anaerobic sludge digestion, they are going to go ahead here. Some anaerobic bacteria digest the bacteria and fungi in the what sludge by producing something called as what gases like what methane, H2S and what CO2, right. These gases forms what the particularly biogas. Now with respect to the what biogas plant only, that itself what we are going to call it as anaerobic sludge digester, where they are going to use something called as what anaerobic bacteria, which actually digest the bacteria and even other organic matter and form something called as what methane. Then again the particularly H2S, CO2 gases can be what formed over here, right? The effluent is what released into the what natural water bodies like what reservoir and streams. Now after all this purification or all this particularly primary and secondary type of treatment, the whatever the water was, that water, the purified water was actually transferred into the what water bodies, right? Like what rivers you consider or streams you consider, right? Then majorly overall steps if you consider here, the primary effluent is actually passed on to the what aeration tank. From the aeration tank where they are going to carry out what constant agitation. Agitation means what mixing, continuously supplying the air over here. Then definitely what will happen, aerobic bacteria are going to grow up. Aerobic bacteria in flux consume something called as what this organic matter, which actually reduces the water, reduces the BOD. Right, from the BOD, what you are going to find out majorly here, the effluent settling tank. From the settling tank, you can find out what flux sediment to what activated sludge. From the activated sludge, small amount of what the particularly sample can be taken, that what we are going to call it as inoculum, passed on to the what the aeration tank. From there, they'll go ahead with respect to the anaerobic sludge digester, where the different methane, then again H2S and CO2 can be produced. Methane can be used for the what particularly as a gas, right? So for the preparation of what food even. Then they'll again go with respect to the what anaerobic bacteria digest the particularly what sludge and produces what the particularly biogas, right? So that what you can find out. Here effluent settling tank, from there the water can be what even transferred to the what natural water bodies. So that like that of what overall the particularly secondary treatment or biological treatment can be what carried out here, right? Then here overally the particularly image of this particularly like what we call it as the microbes, where we can use something called as what to treat this particularly sewage. So this is what the overall image. So this image, whenever you get a time, once you observe it carefully, you will get what all the like what points, and you will understand how actually the purification of what water can be what carried out, or the sewage treatment can be performed, right? So majorly if you consider here, the Minister of what Environment and the Forest has initiated Ganga Action Plan and Yamuna Action Plan to save from the what water pollution. So majorly we can find out with respect to the what some of the our religious places, there also you'll find out lots of what contamination, where people will go to on throwing the what so much of a material inside what the particularly rivers. So to carry out the purification, so there they'll go ahead with respect to the what this particularly sewage kind of water treatment, right? So that what you are going to see. Then we'll discuss something called as what microbes in the particularly production of what biogas, right? How actually this biogas can be produced. Majorly like what this is the structure of what methanobacterium. This is the main bacteria which can be used for the what the particularly production of something called as what the methane gas, right? Majorly if you consider methanogen, right? So this methane bacteria you can find out with respect to the what gut of what ruminant animals, and even it can be known as what archaebacteria, right? Then biogas is a what is a mixture of what gases. Mainly it consists of what methane, right? So produced by the microbes such as what methanogens, right? So methanogens grow anaerobically on a cellulosic material. So cellulosic material means majorly if you consider like what the leaves or different vegetable waste, from there what you are going to get what cellulosic material, onto which anaerobically methanogen is going to grow up and produces what here methane along with what carbon dioxide and H2, right? So example here what they have given a methanobacterium. So this is what the actual structure of what methanobacterium what you are going to see, right? So then methanobacterium is found in the what anaerobic sludge and rumen of what the cattle, like what I have told you, right? With respect to the sludge digestion, there you can find out what they are using. They are actually using something called as what the dung itself. Now with respect to the dung only, what you are finding out, the presence of what methanogenic bacteria. And now from where it came from the what, the ruminant animals what gut, from there itself, like what will we get what methanobacterium. Majorly this methanogen or a methanobacterium is going to help for the what cellulose digestion purpose with respect to the whom, this particularly fodder eating animals, right? So that what you are going to find out. Then biogas is used for what purpose? One is cooking and lighting purpose. Then cattle dung is even used, that is what we call it as gobar, right? Containing contains what, these bacteria. So the dung is used to produce what, biogas or gobar gas, what we call it as, right? Then overally this is what the structure of what biogas you are going to find out. Majorly how they construct it, like consider a concrete tank is prepared, which is of what inside the what the particularly depth, right? So almost what 10 to 15 feet deep they are going to dig it to collect this particularly what bio waste and slurry of what the particularly dung you consider. Then they are going to add a floating cover to it and is placed over the what slurry, and which help on the what rising of as the biogas is actually what produced. So digester will be there, inside the digester only what we are going to add the biological waste. With respect to that only we are going to add something called as what dung or slurry. And what is actually slurry is, this is actually prepared with respect to the what dung and little amount of what water or urine of this particularly ruminant animal can be what used. Then outlet, consider outlet to supply something called as what biogas here, right? Then the outlet to remove something called as what suspended slurry. So here, from here you are going to what remove the suspended slurry, which even can be used as was fertilizer. From this particularly sludge, whatever the content or that content to be what removed, collected, and that can be used as what fertilizer for the what the particularly what plant. So here what they are going to add, the dung and water they are going to add, that is going to get mixed up with respect to the water digester. There the particularly what the overall procedure of what production of what methane can takes place, and that can pass on with respect to this particularly gas holder, and from the gas holder it can reach up to the what all part of the particularly what majorly like what gas where we are preparing the food or even lighting purpose what we are going to use. Here you can consider Indian Agricultural Research Institute, that is what IARI, right? And the particularly Khadi and Village Industries Commission, that is what KVIC, developed a technology of what this particularly biogas production in India. So these are the what two who together itself what developed the technology of what this particularly biogas in India. You should remember that is Indian Agricultural Research Institute, which is there in Delhi, right? And even with Khadi and Village Industries Commission. So both together worked and carried out the production of this particularly what biogas, right? Then microbes as what majorly the biocontrol agent, right? So that what you are going to find out. Now with respect to the biocontrol agent, right, like that they are even going to what control the population of what some of the what even insects too, right? Or insects, some of the like what different purposes. Even we can use it as a fertilizers, even we can use it for as a what something called as what pesticides, like that what microbes plays very important role, right? Then biocontrol is the what use of this biological method for controlling something called as what plant diseases and pest. So plant diseases and pest population can be controlled by means of using this particularly what microbes. Consider example what they have given, ladybird, that is what beetle, right? So controls the what majorly like what aphids population, that is dragonflies control mosquitoes. Now dragonfly controls what mosquito, and this particularly what ladybird or beetle controls the what aphids population. That what you are going to find out. Then majorly you consider chemical pesticides and insecticides kill both the useful and harmful organisms and causes what pollution. But you can find out biocontrol method has no such problems, right? So majorly if we use chemical pesticides or insecticides, that can cause effect to the what the particularly small children's and even so many people, right? So even it is going to harm and causes something called as what loss into the what soil fertility too. So to such kind of what problems majorly like what we can avoid, instead of that we can use something called as what biocontrol methods. So it has what no any such kind of what problems you are going to find out. Right. So majorly if you consider like what Bacillus thuringiensis, majorly this is the biggest example, which is what insecticide, as a insecticide we can use it, right? So which controls the population of what some of the insects which actually feed onto the what this particularly cotton. Now what they have done, we have already discussed it with respect to the what biotechnology application. They have taken a genes from the what Bacillus thuringiensis, added with respect to the what plant, and gene expression is carried out, and this gene expression is carried by means of what agrobacterium tumefaciens bacteria. Now that cry-A-C and cry-A-B or cry-2-AB genes are used to what control the population of what some of these particularly what insects, right? So which actually feeds onto this particularly cotton ball, right? So majorly like what to control the butterfly caterpillars you consider, or the dried spores of what BT available in what sachet right now, right? Then are mixed with respect to the water and sprayed onto the what vulnerable plants such as what brassicas, fruit trees even you can consider, and these are eaten by the particularly what caterpillar. In their gut the particular toxin is released and the larvae get what killed. Like that what this particularly BT or Bacillus thuringiensis is going to act on. So scientists have introduced the BT or Bacillus thuringiensis toxin gene into the what plant, and they formed something called as what this particularly BT cotton. So what BT stands on, that is Bacillus thuringiensis cotton, right? Like this what we can use. Again trichoderma species, mainly like what fungus you consider, that this particularly what trichoderma, they are seen in the particularly root with respect to the what ecosystem. They control what several plant pathogens. So these particularly trichoderma fungi is going to grow up with respect to the what surrounding to the what this particularly root, and controls the particularly what plant pathogens. So plant even can be what disease free here. Then the Bacula virus you consider, especially genus mainly known to be what nucleopolyhedro virus, right? Nucleopolyhedrovirus, or even what we call it as baculovirus, right? So which actually attacks the insect and other arthropods, right? It is going to kill the particularly what arthropods or some of the insects. Like it is suitable for the what species specific, that it is going to be what species specific, and narrow spectrum insecticidal applications you can find out, and desirable in what IPM means integrated pest management program, to conserve something called as what beneficial insects. So it is specifically with respect to the what some of the insects only. So with respect to that integrated pest management scheme, we can use this particularly Bacula virus for killing some of the what specialized what the insects or arthropods which causes what disease or infection to the what particularly plant. Then microbes has what biofertilizers majorly, like how microbes plays very important role as a biofertilizer. So root nodules you consider of what leguminous plant or legumes, you can see the whatever this nodule like structure, right? Then rhizobium is being one of the bacteria, and azospirillum is again one of the bacteria which can be used as what biofertilizers. So majorly what it is, biofertilizers are what the organisms that enriches the nutrient quality of the particularly what soil. So these are the organisms which help up with the growth and development of a plant, and even it increases the what nutrient availability for the plant inside the what soil. So example is what some of the bacteria, fungi and cyanobacteria you consider. So here they have mentioned what rhizobium, which is what symbiotic bacteria in the root nodules of what leguminous plants. So with respect to the what nodules, you can find out the presence of this particularly rhizobium, and it forms what leg hemoglobin, which is pinkish in color, and looks what blood type, so only it is known to be the what leg hemoglobin, which actually fixes what atmospheric nitrogen. So nitrogen availability to the what plant can be carried by means of these microbes. Then free living bacteria in the soil you consider, that is azospirillum, or even what azotobacter you can find out, which actually enriches the nitrogen content of the what particularly what soil. So nitrogen is a major component even required for the growth of this particularly plant. Then there is something called as what mycorrhiza. That mycorrhiza, which is even what symbiotically associated fungi, which example is known to be the word glomus, with the particularly what plant. The fungus gets what food from the particularly plant. The glomus is one kind of what mycorrhizal fungi which will help into the what to carry out or symbiotically associated with respect to the what plant. Then the fungi, fungal symbiont perform the what following functions. Majorly, it is going to carry out what a different functions here, right? So one is absorbs the phosphorus from the what soil and passes it to the what particularly what plant. Then give resistance to the what root borne pathogens and tolerance to the what salinity and drought conditions. So like that of what conditions or the particularly functions performed by means of what these symbiotic organisms, and give overall increase in the what plant growth and particularly what a development. So like this it plays very important role in overall growth and development of the plant, right? Then there is again like what cyanobacteria is one example known to be what the blue green algae. Majorly it is known to be the autotrophic microbe which carries what photosynthesis, comes under what kingdom Monera, known to be what eubacterias. They fixes what atmospheric nitrogen here. Examples what they have given Anabena, Nostoc and Oscillatoria, right? With respect to majorly paddy fields, you can find out what cyanobacteria serve as what the particularly biofertilizer, right? Where in paddy field you will find out water lodging, because these organisms majorly require what moist condition or water content, right? So definitely it will grow up with respect to the what this paddy. It also add organic matter to the particularly what soil and increases the particularly what soil fertility. So that what you are going to find out with respect to the cyanobacteria, right? Now overallly this is what the topic what it was there with respect to the what your majorly PUC second year. So go ahead with respect to these particularly points. Remember some of the points which are actually useful for your CET purposes. So right now, thank you so much. Please do subscribe our channel and do like it and share it. Thank you.